My name is Markus Hartmann, and I'm the CSO and founder of Ciliate, a company from Münster, Germany. And Ciliates, like Tetrahymena, are not only unique organisms for basic research, of course, but also for applied approaches in biotechnology. And that's the reason why we, as a biotech company, are working with Ciliates of the genus Tetrahymena for research and production of protein-based biopharmaceuticals. You can see here an impression of our labs in Münster in Germany. It's the western part of Germany, close to the Dortmund area, where we have at about 870 square meters lab with 25 people. And we have our own production facility here in 900 liter scale, where we multiply and fermentate Tetrahymena. And this is the agenda, as well as also our product pipeline. We are working on ciliate lipase, which are functional enzyme for human use. We're working on a recombinant influenza vaccine called Cyflu from Tetrahymena, which are viral antigens. And last but not least, we are also have a lot of proof of principle studies for biopharmaceuticals, as well as an anti-cancer antibody produced, which has been also published, and you can find this on our webpage. Ciliate.com. I will give a short overview why use Tetrahymena as production host, simply because it's very easy to fermentate, but you all know the characteristics of Tetrahymena. I think it's one of the most important groups of protists. It is commonly found in lakes, ponds, ocean and rivers and soil everywhere in the world. And it's, of course, a eukaryotic microorganism and opposite to a lot of other expression organisms, which are bacterial or yeast bacterial or platforms. And it's, of course, the genome is fully sequenced. It's harmless to humans. It's non-parasitic. It has, very important to note, a biosafety level 1, level for research. It's a contained system, in opposite, for example, to transgenic plants and animals. And we can breed, of course, Tetrahymena without using any animal-based components. That's not so easy for other expression technologies. That's lead us to the development of a biopharmaceutical enzyme for the oral treatment of human exocrine pancreatic insufficiency. You can see here the ribbon band of our lipase in the middle, and here is, the illness is simply pancreatic insufficiency, which is based on pancreatitis. So humans which have pancreatitis needs to swallow normally digestive enzymes from pig pancreas, from slaughter waste. And as alternative, we developed, based on our biotechnological approach, a Tetrahymena lipase, a certain one, in order to replace the deficiency. So we replace the enzyme which is not there by a Tetrahymena lipase, and this is called a replacement therapy, where the enzyme will be then taken orally and digest then the lipids in the gut. So this is our approach, how the production is looking like. We are currently taking an endogenous Tetrahymena lipase, clone this into a transformation vector and transform Tetrahymena. And the vector is a classical episomal-based expression vector. You see here the yellow one is the target gene, the lipase, under the control of an inducible promoter. And we have resistance genes, neomycin, which are working for Tetrahymena as well as for E. coli. So this is a combination vector which we prepare in E. coli and then transform into Tetrahymena, which is, of course, an episomal one, which means that the vector is replicating autonomously. And there we make then clone selection, and after the selected clones with the best producers, we make fermentation and harvest the lipase from the supernatant, not by classical column chromatography that we have done in the past. We are now working with a fascinating new procedure where we crystallize the lipase directly from the supernatant of Tetrahymena. And this works very efficiently. We're adding chaotropic salts, and we get this 100 micrometer long lipase crystals, very pure by centrifugation, which is nearly 95 pure in one step. So this is a fascinating, simple approach to harvest a Tetrahymena lipase, which is very important to make a very short DSP, a downstream process, because of costs, of course. And also we have a very pure enzyme. This you can see here. We have here in the middle, this is a gel. We have reached already a purity of, where's my mouse? Yeah. of 95%. This is the monomer. We have also dimer and trimers. This is not a big issue. This is the gold standard comparator product, which is based on pancreatine. Creon, it's a drug which is available on the market. And this is a pig-based drug which contains a lot of different proteins, and you can see the purity is really high. We have, of course, tested this in an animal model. Animal model which shows us whether our lipase is supporting the digestion. And you see here on the left side, this is an animal model with pigs. And you see here on the left side the so-called coefficient of fat absorption, which is nothing else than, which says nothing else that the fat which animal takes in will be digested and taken up to the gut. So the released fatty acids will be taken up from the fat by digestion. And the animals, green is the healthy condition, a healthy pig, and red here is a pig which has an ill condition, which has a pancreatitis and took a placebo. And you see here on the right side, Creon and Ciliate, which is a ciliate lipase, and both has nearly the same improvement of the fat digestion. So we could prove efficacy based on the same amounts of enzymes which we added. But important to say is here that the purity of our enzyme is much, much, much higher. And important to note also, based on the crystallization process, we received a very dense enzyme with a very high purity and activity, which results in that the drug will be later on. This is the gold standard comparator which is on the market. It's called Creon, and this is the outline of our drug. So the pill burden reduces strongly by this pure tetrahymena lipase, which is an advantage for the patient later on. So this is our lipase drug which we developed. Important to note that we reached already the 7,500 liter scale here. This is a German CDMO contract manufacturer which is fermentating tetrahymena in 7,500 liter scale. So upscale works very well. This is important for pharma partners, of course. And I would switch now over to our influenza vaccine. This is subunit vaccine candidate against human influenza, epidemic as well as pandemic influenza. And we do here nothing else than with the lipase, we express a foreign gene. The case of the lipase was an endogenous gene. I have to correct myself, but this here is a foreign gene. It's an influenza hemagglutinin from the surface of an influenza virus. We take just the gene and clone it to an expression vector. In this case, this is not an episomal vector. We make an integration into the GRL3 gene. So we have a knockout of the mucus cyst protein, and we have then an integral expression in the chromosome. And then we have expression of the hemagglutinin from the virus on the surface of the ciliates. We fermentate the tetrahymena, and then we harvest nothing else than the purified hemagglutinin based on a classical chromatographic cascade and put it in a micelle, or in this case, it was also a nanoparticle, which we received from colleagues from the CNRS in Lyon and at Novartis. And we made mice studies as well as monkey studies together with the CAR in Paris, and mice studies with the University of Siena and the Imperial College in London. These are the results. We could express two different hemagglutinins from different influenza viruses. I don't want to go too much into the details because you can see this also in our publication. We received a very good efficacy in vivo, and not only that, we received a strong immunogenicity after we vaccinated mice with our tetrahymena hemagglutininin, which you can see here on the left side, where we had a strong inhibitory antibody answer. I don't want to go too much to details to all these lines because it says nothing else that we have a strong inhibitory immunogenicity in mice. We tested different doses, and we tested different routes of application, administration, subcutaneously as well as intramuscular, and so on and so on. But all of these approaches worked, and we could show that the lung cells of the mice are not anymore infected. So the lung cells were protected. The mice didn't lose weight, as you can see here, especially with the blue and the black line. And what's very important to note also, subcutaneously as well as intramuscular, our tetrahymena-based influenza vaccine protected the mice against the virus because the virus couldn't proliferate anymore. So the viral load was low in the animals, and this is a very good proof that the combination of our antigen with nanoparticles, but also without nanoparticles, leads to protection. We could also show this in monkeys. And important to note, and this is a more strategic approach here, the tetrahymena-based or the Ciliate-based expression system has advantage compared to other expression platforms, especially in the field of influenza as well as other envelope viruses, that we have a very simple technology. Ciliates can express surface antigens of viruses on its surface because it has a very easily accessible membrane. And this has an advantage towards other technologies which are on the market. You all know, of course, the mRNA technology from BioNTech. We know the vector-based technologies from Moderna and Johnson & Johnson, which are nuclear-based vaccines. But all these technologies are very scattered. They are very complex and expensive. And we are producing here a subunit vaccine in a very simple approach, which is much more cheaper than this mRNA and vector-based vaccines. And the other technologies which are on the market, as you can see here, these are protein-based subunit vaccines from Novavax, for example, or from Protein Science now called Sanofi. They produce in insect cells, and the insect cells are very complicated because transformation with insect cells works with baculoviruses, and tetrahymena is easily transformable without viruses. So just a transfection takes place with electroporation, which we do in our labs, and that makes it very easy and easy to also to upscale the process, because for insect cells, you have to upscale an insect fermenter up to 10,000 liter, and then you have to add the same amounts of baculoviruses to the fermenter. And that's not the case for tetrahymena. Last but not least, the egg-based technology and the human and monkey tumor cells are very outdated because they breed simply viruses. So we think this is a very simple approach to produce vaccines, and we have transferred also the tetrahymena expression technology to a lot of other enzymes, influenza vaccines here, but also malaria surface antigen, and last but not least to anti-cancer antibodies. We have expressed also a monoclonal antibody, a therapeutic monoclonal antibody called rituximab in tetrahymena, and could prove efficacy ex vivo. And we have meanwhile expressed 22 different proteins, recombinant proteins in tetrahymena, and this I think is a very good proof that tetrahymena as a ciliate is a very good expression technology. Of course, we are looking for collaborations in the field with our colleagues from the basic research, and especially promoters and resistant genes are of special interest for us, because we always need to have better promoters and better resistant genes, but also other approaches. I heard that tomorrow there will be a Turkish group showing some approaches about proteases from tetrahymena. This is, of course, also very interesting. Thank you very much.